Today, I will talk about cognitive and behavioral genetics. First, I will introduce the relationship between cognition and behavior. When we receive the environmental stimulus, the brain will operate a complex cognitive process, then establish knowledge base or react. This process is influenced by the neurotransmitter and genes. There are close connections among these components. The cognitive genetics is to study the influence of genetic variation on cognition, and behavioral genetics involves the use of genetic methodologies to study the nature and origins in the individual differences in behavior. Behavioral genetics is often associated with the nature versus nurture debate, that is, which one does matter on behavior, genes or environment? This micro course will introduce some studies of human cognitive and behavioral genetics. In many cases of cognitive and behavioral genetic study, it wouldn't be ethical nor feasible to do true experiments. Traditional twin studies mimic some key feature of an experiment. Scientists take advantage of natural occurrence, identical twins, to understand whether or not genetics influences individual differences in a trait. Another traditional method, adoption method, then offers a way to investigate the environmental impact. By measuring a trait in large groups of twins and siblings, we can understand the effect of gene vs environment on a trait without looking directly at people's genes. With a complete mapping of the human genome, genome-wide association studies, aka GWAS, began to proliferate. In a GWAS, associations between single nucleotide polymorphisms, aka SNPs, and human diseases or traits are assessed across the genome to determine whether any variant is associated with the trait of interest. Galton first introduced the twin methodology in 1875. Twin studies are based on the existence of two types of twins, monozygotic and dizygotic. Dizygotic twins are like ordinary siblings to share on average 50% of their genetic material. Twin studies compare these types of twins. If genetics is important, then monozygotic twins should be more similar than dizygotic twins. We can also assume that any difference in traits between identical twins may come from the environment. Now we add a new group, adopt siblings, who don't share genetics but share their environment. If genetics is important, then monozygotic and dizygotic twins should both be more similar than this new group. If the environment is important, then they should be roughly equally similar. Let's see one study results of these three groups of IQ trait. In these figures, each dot is a pair of twins or adopt siblings. The x-axis represents the IQ score the first twin or sibling got, and the y-axis represents that of the other one got. Then we compare how similar the pairs were, and this can be observed by how close the dots are to the orange line in the middle. If their measurements are very different, their dot will be far from the orange line, and the correlation coefficient R is a statistical measure of the strength of the relationship between two variables. R value of 1 indicates a perfectly linear relationship. In the adoption group, it's much less than the twins group. Genetics appear to be important, However, it is not zero, and for monozygotic twins, it is not 1. Something about their rearing homes is shaping some similarity in IQ. Let's see another study aims to discover whether antisocial behavior inherited. The sample of adopted children was classified into 2 groups, whether or not their birth parents had a history of antisocial or aggressive behavior. The x-axis refers the adopted home. The value of 3.5 means high-risk home. The value of minus 1.5 means more nurturing and protective environment. The y-axis refers the number of adoptees who had aggressive behaviors. The solid and dashed lines represent whether or not their birth parents had antisocial behaviors. The solid line tells us whether or not there is a genetic effect depends on the environment where they are reared. If they were reared in the nurturing, protective environment like minus 1.5 of X value, for example, the genetics didn't matter. There is a model of psychopathology called the diathesis stress model. If you inherit the diathesis, you are inheriting a genetic vulnerability. That doesn't mean necessarily you will get the disorder. In the past few decades, without the technique of DNA sequencing, studies are proceeded by a very crude way. In twin study of alcoholism, for example, the percentage of monozygotic twins are alcoholism is higher than that of dizygotic twins. So we suggest that gene has a great influence on alcoholism. Nowadays, we finally found the key answer to alcoholism, thanks to the DNA sequencing technique. When we drink alcohol, we need enzymes to transfer ethanol to acetaldehyde and then to acetate acid. However, if the gene that makes this enzyme, ADH, happen a SNP, the function of alcohol metabolism will be impaired, and we will feel uncomfortable, which can stop people from getting alcoholism. Many Han Chinese have the SNP of the gene ADH, while few have it in Western countries. Therefore, there is a higher percentage of alcoholism in the West. Last but not least, environmental factors may have little influence. According to the studies of behavioral genetics in the past, scientists proposed 4 laws in behavioral genetics. First, all men behavioral traits are heritable. Second, the effect of being raised in the same family is smaller than the effect of genes. Third, a substantial portion of the variation in complex human behavioral traits is not accounted for by the effects of genes or families. Last, a typical human behavioral trait is associated with very many genetic variants, each of which accounts for a very small percentage of the behavioral variability.